Dear students, now the next application of the bioinformatics is the gene therapy. Now, how it is useful? So actually, this is the in the this development is in juvenile stage, and gene therapy approaches uses changes in the expression of genes of the treatment of disease. So, however, the clinical trials for several different types of cancer and other incurable diseases are underway. The first, gene therapy. Then fourth one is the antibiotic resistance. So in antibiotic resistance, what happened? So E. facilis is a pathogen and causes a bacterial infection among the hospital patients. By utilizing the bioinformatics, it was revealed that it contains a virulence region made up of number of antibiotic resistant genes, and that region is called as pathogenicity island. So if we analyze that sequence, that could provide useful markers for detecting the pathogenic strains, and in the future could be helpful to establish controls to prevent the spread of infection. Now, then the agroinformatics. The application of bioinformatics in the agriculture science, including plant genomes, is termed as agroinformatics or agriculture informatics, and it concentrates on the aspects of sequencing on of the benefits for the agriculture community. So same thing here, we have to apply it for the plant, the sequencing, gene analyzing, and which gene is responsible for which fruit. So it will build healthier, more disease-free resistance plants we can produce. Then phylogenomics and evolutionary studies. In this, phylogeny is the branch of science dealing with the origin and evolution, and in the past we were studying phylogeny evolution merely on the basis of morphological and anatomical features. But here, if we know the genome sequence, we will very well, by applying the karyotype, studying of karyotype, we can know how these phylogenics and evolutionary studies are carried out, now how evolution is taking place. So this evolutionary on gene basis, basic level. Then the creations of bioweapons. So this is slightly danger, but by manipulating the gene, we can make a bioweapon. What is this actually? Although bioinformatics have so many useful applications, but it is also true that there may be a harmful aspects interwoven with useful things. After genomic data of pathogenic species is available on internet, it can be utilized in the information of bioweapons. This virus poliomyelitis has been built by scientists using entire artificial means. US Department of Defense have funded the research as a part of biowarfare response program, and they want to prove that reality of bioweapons to the world. However, such practice must be discouraged for benefit of mankind. And nowadays, this COVID-19 virus is a bioweapon. It is created in the lab, and now it is very harmful to the humankind. So we are facing the same problem. So how it is possible that destructed gene is created by using the bioinformatics from the gene, and that virus is created, and that virus is now proved fatal to the humankind. Then literature retrieval. The most important application of bioinformatics is the literature retrieval. That the scientists, researchers are using the facility for designing their problem, what part of research work had been done, documented similarly themselves throughout the globe for the comparison of data and publishing. So they use this data for their research. Lastly, the genomics is used analyze the entire genetic complement of a species. That is, proteomics is the study of location, structure, function of entire protein content of the cell or body. During the last decade, bioinformatics has become the powerful tool to explore the treatment of genetic disorders, utilizing the genomic approach as well as the level of proteins to obtain accurate three-dimensional structure for all known protein families, protein domains, and protein folds. So this is the use of bioinformatics to the humankind. But bioinformatics are there, but for that we require the bioinformatics database. So when database is there, then it is possible to build a bioinformatics center for to understand the or to get the knowledge about these genes and all these things. So this is called as bioinformatics database. Now, databases are convenient system to properly store, search, and retrieve any type of data. So actually, it is a knowledge bank, and we if we study, do the research, we have to keep in a good condition, and that is called as database. A database helps to easily handle and share large amount of data and supports large scale analysis by easy access and data updation. So this is actually to get the knowledge about all these bioinformatics or about the gene or the protein. So diverse type of information may be stored in biological database. For example, sequences of DNA, RNA and protein, images of 2D gel, structures of macromolecule, organism specific information, metabolic pathways and information from the literature. So this is the database, bioinformatics database. Now, primary database. These are the primary repositories of data used to store the nucleic acid, protein sequence, and structural information of biological macromolecule. So, this is the primary database. That is NCBI, GeneBank, DDBJ, Swissport, PIR, PDB are the names of some primary data repositories. So, they put the sequences of all these macromolecules and the genes. Primary. And then secondary what? The secondary database, this database contain the additional information derived from the analysis of data available in primary repositories. So secondary database are analyzed in a variety of ways and contain different information in different formats. So this is the secondary database. Then composite database. A composite database combines information from various primary databases and make it too convenient to search the desired information without querying to all these primary databases. So this is the composite database. Then nucleotide sequence database means nucleic acid. So gene bank, EMBL, nucleotide sequence database and DNA data bank of Japan are the major sequence repositories from which various databases have been derived, and these serve as a primary database. Means they have analyzed whole DNA sequence of the human being and they put the information on the in the database. Now it is available. Gene bank. Gene bank is the most comprehensive and annotated collection of publicly available DNA sequence is a part of International Nucleotide Sequence Database. We know that now whole human genome is analyzed. In the same way, the next is that protein sequence database. So protein has a wide range of database. Each has its own usefulness. The most important protein resources available are. So in the same way, like nucleotide sequence, the protein sequence is also analyzed for different various enzymes. And that database is available from the following databases terminals. For example, Swiss protein sequence database, then translated EMBL, then UniProt, prostate, structural database. So from these various agencies, we get the knowledge about the protein sequence. That is first one, that is Swissport is database of created protein sequence and provides high quality annotation with the minimum redundancy. And Swissport is a cross-referred with several other databases including nucleic acid and protein structure database. The second one, translated EMBL. Translated EMBL translation of EMBL in nucleotide sequence database was created in 1996 with the objective to fill the gap between the flow of genomic data and annotate protein sequences. Means whatever the nucleotide sequence, we have to see which protein is created from that sequence. So that database is kept here. Then protein information resource, PIR. The Protein Information Resource, established in 1984 by the National Biomedical Research Foundation, is an integrated public bioinformatics source which maintains the protein sequence database. So, in this way, this sequence knowledge is put on the best kind of human being for the research. Then UniProt, National Institute of Health, in 2002 provided funding to PIR along with its collaboration European Bioinformatics Institute and Swiss Institute of Bioinformatics to contrast universal protein resource, means protein sequence they prepare. Then ProSite. The ProSite database consists of protein families, domains, and functional sites which serve as a biological signatures explained as a patterns or profiles, and this valuable biological information on domains, protein families, or functional sites is provided by documents associated with all signature present in the database. So they keep the protein database. So I think now in continuation with this, the structural databases are there. Structure means three-dimensional structure of biomolecule plays an important role in their functions and maintenance of the structural features of an organism. So that structural database is available. Number one, I'm not reading this, only I just give the names. That the Protein Data Bank, it is established in 1971, is an international structural database which contains information about three-dimensional structure of biological molecules like proteins and nucleic acids. So it is related to structure. The second one is the molecular modeling database. NCBI also maintains the structural database of biomolecules. It is in 3D format. The nucleic acid database. The nucleic acid database is a comprehensive relational database devoted to three-dimensional structure information of nucleic acid. So it is very easy if we get the 3D structure of any molecule of protein or nucleic acid. Then fourth is structural classification of proteins. That is, the structural classification of protein database was created in 1995 by Murzin et al. It is maintained at Cambridge with aim gather information about the structural similarities of protein to increase our understanding of protein evolution and development. So they keep the maintain the Cambridge structural similarities of different proteins. So this is very important. And lastly, the class architecture topology homology. So this CATH database established in 1993 is a protein structure classification based on four levels, namely class, architecture, topology, and homology. CATH contains hierarchical domain classification of protein structure present in Protein Data Bank and is maintained at University College, London. So these NMR structures along with the crystal structures with resolution of 4 angstroms or better than these have been considered while deriving the CATH. So actually this will keep very detailed little structure or microstructure of the protein and nucleic acid. So this is now available for the research, and this is the because of the bioinformatics and bioinformatics database. Nowadays, it is available for the researchers. Complete human genome is now analyzed and complete protein structures and its 3D structures are available for the researchers. So next time we will see the next topic, that is the biostatistics. Thank you.